Yes, this is 300 plus academy where all we do is exams tutoring, ensuring that you ace your next exam. Still on the subject matter of how substances move in and out of the cell, or what we call transport across cell surface membrane. I haven't looked at osmosis. Let's quickly look at the differences between diffusion and osmosis. When you talk about diffusion, it involves movement of solid molecules, gaseous molecules, and liquid molecules. When you talk about osmosis, it involves only movement of water molecules, as you can see on your screen. You don't need a semi-permeable membrane for diffusion. You need a semi-permeable membrane for osmosis. Talking about diffusion, molecules move into any space accessible to them. In the case of osmosis, water molecules move between two solutions of different osmotic pressure, leading to establishment of isotonic solution, that's osmotic equilibrium. When you talk about diffusion, it occurs in both living and non-living things. When you talk about osmosis, it occurs only in living things. That ends that on the differences between diffusion and osmosis. Okay, let's quickly look at an experiment to demonstrate osmosis using yam cavity, that's a living membrane, or let's say something like pawpaw. So what happens is that this here is a trough of water, trough of distilled water, trough of distilled water. So two yam cavity, with enough space drilled into the middle, is suspended in a trough of water. One of the yam, let's say yam A now, is filled with a strong salt or sugar solution. This is strong salt or sugar solution. This is a strong salt or sugar solution, is poured into A, whereas yam cavity B is left with no salt, no sugar, with nothing. That is the control experiment. And of course, in most biological experiments, we usually talk of a control experiment to show that something is not going to occur when certain things or when certain conditions are absent. So this is our control experiment. So a strong salt solution is poured into the cavity of A, and both yam are then allowed to stand for some hours. After some hours, it is found, okay, let L1 be the level of the strong salt solution. After some hours, the level of the solution in yam cavity is found to have risen. And the reason for that is very simple. Here is a hypertonic solution here. In the trough, what we have is pure distilled water, that is hypotonic solution. Osmosis had happened here such that water had been drawn in. Now the yam here is the living membrane, is the selectively permeable membrane. So water had moved in from region of what? Hypotonic solution to a region of what? Hypertonic solution through a semi-permeable membrane. And that is why the level of the water here and the level of the solution had risen. In yam cavity B, water does not move in. It remains the way it is, telling you that osmosis had not happened here. So that's that about this experiment, telling you that whenever one region has a higher osmotic concentration or osmotic pressure, then osmosis is going to proceed. Let us look at the importance of osmosis in plants and animals. The first one, as you see on your screen, you can see a diagram showing the root of a plant and with adjoining cells. And of course, you can see some arrows showing the direction of the movement of water. Normally, you have soil water and then you have the root hairs. I need you to understand that water is going to move via the root hair into the plant cells via osmosis. That's we're talking about water and mineral salt. Like I wrote here, I said absorption of water into the vacuole of the root hair is by osmosis. The water then moves from the root hair to the cells also by osmosis. Simply, osmosis ensures continuous absorption of water and mineral salt, uptake of water from the soil into the root air and then consequently into the cells of plants is by osmosis. So it is osmosis that ensures absorption of water. Opening and closing of the stomata by the guard cell is by osmosis. When the guard cells become turgid, the stomata opens up. When the guard cells become flaccid, when they are not turgid, when they become flaccid, then the stomata closes. That is that about that. You can see that as well on your screen, talking about opening and closing of stomata. When the stomata bulges, when it becomes turgid, the stomata opens up. But when it becomes flaccid, then the stomata, closes up. Also, talking about osmosis, osmosis gives turgidity to the plant cells and consequently supports. In fact, one of the UTME questions says, when a parenchymal cells of plants, when they are turgid, what role do they perform? They perform the role of support. When plant cells are turgid, when they are filled with water, when water has moved into them by osmosis, then they become turgid, performing the role of support. That is why when you see leaves of plants that are taken, that's been soaked in water, that has been placed and allowed to stand in water, when you, after some time, when you bring it out, you see that the leaf is not going to sag. What the leaf will do is that it will be in a flat horizontal position because it is turgid, because the cells have taken in water and they are now turgid. Now they are performing the role of support. But when you see a leaf that has not been allowed to stand in water, what happens is that it becomes flaccid, it falls off. It's not able to stand in a flat horizontal position. So when a plant cell are taking up water by osmosis and have become turgid, that turgidity gives support to the plant cells and then the plant as a whole. Then in animals, intracellular movement of water is by osmosis. That's within the cell. Movement of water is usually by osmosis. Is one region having a high concentration? Is the other region having a low concentration? Thereby, once there are changes in osmotic concentration in various parts, then movement of water is bound to occur by osmosis. So intracellular movement of water in animals is by osmosis. Also, reabsorption of water in the kidney tubules. Now, during the formation of urine, let's say the nephron, we have the first convoluted tubule where most of the water is reabsorbed, and then you have the loop of Henle, where, of course, the longer the loop of Henle, the more the water reabsorption. So when you look at the kidney tubules or the nephron, generally water is being reabsorbed so that we maintain the right amount of water in the body, and much water is not lost in urine. So that's talking about osmoregulation, reabsorption of water in the kidney tubule so that the water doesn't go into urine is via osmosis. Also, from undigested food materials in the large intestine, undigested food materials in large intestine, instead of passing out the undigested food materials with enough body fluid in them, with enough water in it, when the undigested food material, fecal material, is allowed to stand in the large intestine for a number of time, then water in it is going to be absorbed back into the body. So this is just talking about conservation of water in the body. So absorption of water from undigested food materials in the large intestine is by osmosis. Also, osmosis gives turgidity to the animal cells. That said, hemolysis, that's the bursting of the red blood cell, as we've stated previously, is by osmosis. Crenation is also by osmosis. So these are just some few instances of osmosis in plant and animal cells. Now, worthy to note, take note of this, that when you talk about absorption of water absorption in plant cell, water absorption is by osmosis. But you see, the mineral salts moving along with the water, the movement of the mineral salt is by diffusion. This is a UTME question. Very important. Take note of this. Take note of this, and take note of this as well. I thought about turgidity here in plant cells. The turgidity, you know, when a plant cell, let's say a leaf, is allowed to stand in water for some time, obviously it should have taken up water by osmosis. And when that happens, what happens to the leaf is that it is able to stand in a flat horizontal position. It is not going to sag. So if it's in a flat horizontal position, the reason is because the cells are taking up so much water now, and as they become turgid, now they are performing the role of support. So when a plant cell, you know, is turgid, it performs the role of support. Now, also take note that we said water absorption is by osmosis from the root air to the cells. But you see, the mineral salt that comes along with the water is moving into the root air of plant cells by diffusion. So take note of that. Water absorption is by osmosis. Uptake of mineral salt is by diffusion. Now, I haven't dealt very well with osmosis. Let's look at osmosis in plant cells. What we want to talk about here is turgidity and what we call plasmolysis. We want to quickly look at turgidity and plasmolysis. That's the essence of trying to look at how osmosis happens in plant cells. So usually when you talk about a plant cell, you know, you're going to have the cell wall, the cell membrane, and then you have the vacuole on the inside. The vacuole is surrounded by the tonoplast. So the thing is this, the osmotic concentration of the protoplasm, of the inside of the cell, from the vacuole to the cytoplasm, is higher than what you have in the surrounding of the plant. So usually we say the inside of the plant cell is hypertonic. The inside of the plant cell is hypertonic, while the outside, let's say the soil in which it grows and all of that, is hypotonic. So the result is that according to osmosis, water will move from hypotonic solution to a hypertonic solution. The moment, so in this regard, we expect water to move into the plant cell, you know, especially to the cell sap, to the vacuole, what we have at the center. So water is going to move in by osmosis, and that's what I mean by saying plant cells have a higher solute concentration. That's what I mean by saying plant cells have a higher solute concentration than that of their surroundings. The solutes are located mainly in the sap vacuole, which is surrounded by two membranes. That's the plasma membrane on the outside, and then the tonoplast, which surrounds the vacuole. So plant cell plus a weak solution, that's hypotonic solution here, means that water is going to be drawn into the plant cell. That's water moves into the vacuole by osmosis. That's what it means. Water is going to move into the vacuole by osmosis. Now, the moment water begins to move into the vacuole by osmosis, the plant cell swells, and the content of the cells, you know, is being pushed as against the cell wall, because as water moves in, the vacuole is going to swell, and as the vacuole swells, it presses the organelles and the content of the cell as against the cell wall, exerting some kind of pressure. Now, that pressure being exerted as against the cell wall of the plant cell is what we call turgor pressure. You see, as a plant cell takes up, look at it, as a plant cell takes up water by osmosis, the protoplasm presses against the cellulose cell wall, and an internal pressure is developed called turgor pressure. So as the content of the cell begins to press against the cell wall as water comes into the cell, the cell continues to swell. You see, I said here the plant cell swells. So does it mean that the plant cell is going to continue to swell indefinitely? No. As the plant cell swells, a stage is reached where the cell wall cannot expand again, cannot accommodate more swelling. At that point, we talk of a full turgor, or we say the plant cell is fully turgid. At this situation, what it means is that the plant cell cannot take up more water. It means that the cell wall cannot stretch further. You see, I said plant cell but does not burst because of the cellulose cell wall resists further expansion. If this were to be an animal cell, if this were to be an animal cell with no cell wall but only cell membrane, you know, with only cell membrane, the cell is eventually going to burst when placed in a hypotonic solution, leading to what we call hemolysis. But because this is a plant cell, the plant cell is going to swell. Turgidity or full turgor is going to be created in the plant cell, but the cell wall is going to resist the turgidity and is not going to stretch further, and that will prevent bursting, creating what we call turgidity in plant cell. But if this were to be an animal cell, it does not have cell wall. The cell membrane of the cell cannot withstand that pressure, so the cell is going to burst, leading to what we call hemolysis. But we are going to look at osmosis and animal cell much later. The important thing is that the cell swells but does not burst because the cell wall resists further expansion. So when the cell wall cannot stretch again, we talk of full turgor or turgidity. And when we talk about turgidity, I repeat, turgidity plays the role of support and maintaining plant shape. Now, what that means is this. Imagine we have a leaf and we allow the leaf to stand in water for some time. So it now means the cells inside the leaf, they are hypertonic, while the water on the outside of the leaf is hypotonic. Water will be drawn into the cells of the leaf, specifically the parenchymal cells of the leaf. The cells of the leaves, they become turgid. And when a cell is turgid, as you see in that diagram, you can see, when a cell is turgid, what it does is that it performs the role of support and maintain the shape of the plant. If you allow a leaf to stand in water, you see that the leaf now, if you bring it out of water after some time, it's going to stand in a flat horizontal position. But if it were to be a leaf that has been out of water for long, you see the leaf is going to stand because the cells had not taken up water, had not become turgid, so they cannot perform the role of support. So turgidity plays the role of support support and maintaining the shape of plants. So that is turgidity for you. It is when we allow a plant or a plant cell to stand in a hypotonic solution such that water is now drawn into the cell by osmosis. But take note that I said the cell does not burst because, of course, it has a cellulose cell wall, a rigid cellulose cell wall, which is able to resist the turgor pressure. Look at what we have on our screen. You can see that diagram. Cell placed in external solution whose solute concentration is lower is hypotonic. You can see water enters the vacuole by osmosis. The protoplasm expands, turgidity increases. You can see when we reach full turgor, the size of the plant is now very big, and that is what turgidity is about. Turgidity plays the role of support. In the second case, talking about plasmolysis and or what we call flaccidity, which is going to be shrinkage in this time around. If a plant cell is allowed to stand in a hypertonic solution, so let's say we have a plant cell, the inside of the plant cell is hypotonic, the outside is hypertonic. So now means water is going to move out of the cell into the surrounding. In this case, as water is drawn out of the vacuole of the plant cell by osmosis, what is going to happen is that you see that the cells begin to shrink and the cytoplasmic lining from the cell wall is pulled inward. The cytoplasmic lining is pulled to the inside. You can see that incipient plasmolysis. When the protoplasm continues to shrink, turgidity is decreasing And you see a stage will be reached when we are going to have full plasmolysis, where all of the internal content of the cell is going to be rounded off into a ball. That is flaccidity. It is the shrinking of the vacuole and pulling away of the cytoplasmic lining from the cell wall. Talking about plasmolysis, as I've shown you, if you allow a plant cell to stand in a hypertonic solution, according to osmosis, water must move from a region of hypotonic solution, that's from a region of lower solute concentration, to a region of higher solute concentration. So water is going to be drawn out of the sap vacuole. That's why I said plant cell placed in a hypertonic solution, which is on the outside. It means water is going to be drawn out. And when water is drawn out, it means that the content on the inside of the cell, that's the protoplasm, the protoplasm of the cell, is going to shrink. And then the cytoplasmic lining of the cell is going to be pulled away from the cell wall, such that, as you can see in that diagram, the whole content of the cell is going to be rounded into a very small ball. That's talking about full-blown plasmolysis. So when you talk plasmolysis, it happens because the plant cell is placed in a hypertonic solution, and it is the shrinkage of the protoplasm and pulling away of the cytoplasmic lining from the cell wall. Look at that diagram on your screen again, and that is that about osmosis in relation to plant cell. If you place in a hypotonic solution, you talk of turgidity because water will be drawn into the sap vacuole, into the interior of the cell. If you place a plant cell in a hypertonic solution, then water is going to move from the cell sap, is going to move from the cell, and is going to move out. The whole interior of the cell, the protoplasm, is going to shrink, is going to be rounded off into a small ball in a process called plasmolysis or flaccidity. Okay, now let's look at the osmosis and animal cell. As you see in the diagram on your screen, we have about three situations there. The one at the center is when the red blood cell is isotonic with the plasma in which it is suspended. We know that normally the red blood cells are suspended in the plasma. So normally the red blood cells and the plasma, they are isotonic. That's they are at osmotic equilibrium. So what happens is that in this case, the cell maintains its shape because the rate at which water leaves the red blood cell equals the rate at which water moves into the red blood cells at this stage. So we don't have a problem with that. But you see, when a red blood cell or an animal cell is left to stand in a hypotonic solution, that's in a solution with low osmotic pressure, or what we call hypoosmotic solution. Recall that osmosis is movement of water from a hypotonic solution to hypertonic solution. So if a red blood cell is left to stand in a hypotonic solution, as you're seeing in that diagram, the meaning is that water will move from that solution into the interior of the cell, into the cell, which is having a higher interior concentration than what is on the outside. So water will move into the cell. This cell in this case continues to swell. It continues to swell as more waters continue to come in. But you recall that this is an animal cell, no plant, no cell wall, no cellulose cell wall. So it gets to a stage where the cell membrane here cannot tolerate more swelling. So what happens is that the cell membrane eventually bursts. So the cell eventually bursts open because the cell membrane cannot resist the turgidity. Hemolysis is the bursting of the red blood cells in the plasma. I said normally the blood plasma and red blood cells are isotonic, that they are both at, equilibrium, talking osmosis. That is, they have the same osmotic concentration. So if for any reason the osmotic concentration of the plasma falls such that the plasma now becomes hypotonic to the inside of the red blood cells, then it means that water will now move from the plasma and move into the red blood cells by osmosis. Continuous absorption of water, that's if this situation continues such that water begins to move from the plasma into the red blood cells, will make the red blood cells fully turgid and it will eventually burst. Why is it going to burst? Why is it that it cannot resist turgidity as we have in the case of plant cell? It is very simple. The cell membrane is thin and cannot withstand the turgor pressure. So the cell will eventually burst. So the bursting of the red blood cells, when the red blood cells is placed in a hypotonic solution, in this case, the solution is the plasma, is what we call hemolysis. So since hemolysis leads to bursting of the red blood cells, so we say hemolysis leads to anemia, which may eventually cause death if not checked. You know, when we say anemia, anemia talks about shortage of red blood cells. So since hemolysis is leading to the bursting of the red blood cells, it now means if hemolysis occur and a significant amount of red blood cells had actually burst, that is going to be what? Anemia, and if not checked, may cause death. Also, recall that animal cells have no cellulose cell wall to resist turgidity, hence the bursting of the red blood cells in the plasma. Now, let's look at the reverse situation. If that red blood cells were allowed to stand in a solution with a high salt concentration, that's in a solution that is hypertonic to the inside of the red blood cells. So in this case, it means the inside of the red blood cell is hypotonic and the outside, that's talking about the plasma surrounding the red blood cell, is hypotonic. So the implication of this is that water immediately begins to move from the inside of the cell into the surrounding solutions. You can see hypertonic solution, conch saline. So as water is being drawn out of the cell, the red blood cell here is going to shrink, is going to shrivel. Look at it. If the osmotic concentration of the plasma is higher, that's hypertonic, water leaves the red blood cell by osmosis. The red blood cells will shrink. The shrinking of the red blood cell is what we call crenation. Is what we call crenation. Frequent use of laxatives or excessive intake of salt can actually lead to crenation. When we say laxative, we're talking about things like Epsom salts, that's brand of magnesium sulfate, heptahydrate. Sometimes people take this to aid digestion and all of that. Constant use of this, it means that this is going into the plasma, increasing the osmotic concentration of the plasma, making the plasma hypertonic to the inside of the red blood cell. This will then mean that water will be drawn out of the cells. So if this persists, it simply means the red blood cells will shrink, a situation we call crenation. As you can see on your screen, you can see the shriveled red blood cell. It is no longer biconcave in shape, but it is now, rough. So that ends that is osmosis as it affects animal cell. Now, let's talk about active transport. Recall that I had said that active processes is going to consume cell ATP. That's the energy currency in the cell. Okay, look at the diagram on your screen. Let me quickly talk about sodium potassium pump as it relates to active transport. In fact, more than a third of all the cell ATP is used for sodium potassium pump. Naturally, the amount of potassium on the inside of the cell is more than the amount of potassium on the outside of the cell. Yet, potassium is still being pumped from region of low concentration outside the cell to the inside of the cell. And then talking about sodium, the amount of sodium that is on the outside of the cell is more than what we have inside the cell. Yet, sodium is still being pumped out of the cell to the outside. All this talking about sodium potassium pump from region of high concentration to low concentration is about action potential, maintaining the difference in charge between the inside of the cell and the outside of the cell. For every 2 potassium that comes into the cell, 3 sodium moves from the inside of the cell to the outside. And the movements are both from region of what? lower concentration to region of higher concentration. Both movements are uphill movement. Both movements are as against concentration or diffusion gradient. As you can see that diagram, you can see how sodium binds to at least 3 sites on the membrane protein there, and it's been taken outside as to the extracellular. And you can see the green triangular potassium to binded to its site on that membrane protein and being on the inside. So for every 2 potassium that comes to the inside of the cell, 3 potassium goes out, so that the electrical difference between the inside and the outside of the cell is maintained. But I like us to know that movement of sodium from the inside to the outside is from the region of lower concentration to a region of what? Higher concentration on the outside. And the movement of potassium from the outside of the cell to the inside also, it's from a region of what? Lower concentration to a region of higher concentration. This is a perfect example of active transport, and in this case you see in that diagram that adenosine triphosphate APPP, you can see that there, is being utilized. So that is it. When you talk about active transport, that's just one case out of so many. When you talk about active transport, it is the energy consuming transport of molecules or ions across a membrane against concentration gradient, that's uphill from region of lower concentration to a region of higher concentration. Energy is required because the substance must be moved against its natural tendency to diffuse in the opposite direction. The way substances wants to diffuse naturally is from a region of high concentration to low concentration, but this time around the molecules of the substance are being moved against their natural passive tendency, and that is what we call active transport, moving molecules from region of lower concentration to higher concentration. That's uphill movement of molecules, this time consuming energy. So when you look at it, ATP comes from respiration. So without respiration, where is the cell going to get energy for active transport to occur? You see, so without respiration, active transport is not possible. Look at it. I said the other time that diffusion can occur in both directions, that is, diffusion is reversible. In the case of active transport, it is not reversible. In the case of active transport, it is in one direction. It occurs in one direction. It is unidirectional, unlike diffusion, which is reversible. Now, energy is supplied in form of is supplied in form of ATP made from respiration. Thus, without respiration, active transport is impossible. Now, take for instance, in the case of seaweeds, which takes up iodine so vigorously that it is more than 2 million times concentrated on the inside of that seaweed than you have in the surrounding water. Yet, the little iodine that is still on the outside, the seaweed still continues to take it up. That is a clear example of active transport. Just like your facilitated diffusion, it requires a membrane protein, it requires a carrier to move it across. You need to understand that active transport also requires highly selective protein carriers within the membrane to carry the molecules against its natural tendency. And talking about active transport is very important because it allows a cell to take up more molecules even when it already has enough of that molecules on the inside. Look at it. Active transport is an important function of any cell as it enables it to take up additional molecules of a substance that is already present in its cytoplasm in higher concentration. Take for example, the inside of the cell is very rich in potassium already, yet via active transport, two atoms of potassium, 2 ions of potassium still will move into the cell. Look, the inside of the cell already is rich in potassium, so 2 ions of potassium will still come from the outside, from region of low concentration, to move to the inside of the cell. Talking about sodium ion, the outside of the cell is already rich in sodium ions, yet the few sodium ions that is present on the inside of the cell will still move from region of lower concentration on the inside of the cell to the outside of the cell. So for every 2 potassium ion that comes into the cell, 3 sodium moves out of the cell. That's what we call the sodium potassium pump, and it requires energy. Okay, another example is the liver cells. You know, when we have excess glucose in the plasma, it is the function of the cells of the liver to take up glucose and convert it into storage as glycogen. So look at it. The liver cells are already rich in glucose, yet they will still continue to mop up glucose in the blood, even from region of lower concentration in the plasma of the blood, taking it up into the liver where it is in high concentration. Look at it. For example, liver cells will be unable to accumulate glucose molecules from blood plasma, as glucose concentration is often higher inside the livers than inside the liver cells than it is in the plasma. So these are clear examples that, look, active transport is a very important process that occurs in living things. So talking about active transport is the energy consuming transport of molecules against concentration gradient. It requires a protein carrier. And talking about active transport, it is unidirectional, taking place in one direction only. So that is just what active transport is all about. Yeah, moving on, as you can see on your screen, the next thing is talking about bulk transport of materials in and out of the cell. You can see the diagram on your screen talking about phagocytosis, more or less exactly what we call cell eating, as those solid molecules had landed on the cell membrane. The cell membrane had invaginated, forming a vesicle. That's what I talked about here, to form a vesicle. That's an example of cell eating. And in this case, the cell is also going to use its energy to make this to happen. And then looking at the second diagram you can see on your screen, known as pinocytosis, you can see the particles in this case are a little, they are even smaller and more, microscopic than what you have in A. This is called cell drinking, taken up in form of liquid. You can see what a bulk transport of materials into the cell occurs by an infolding, occurs by what we call, when you say infolding, it's the same thing as saying invagination. Invagination or extension of the cell surface membrane inward to form a vesicle. That is what bulk transport is about. That's what endocytosis is about. Intake. Now, the endocytosis are of 2 types. We have phagocytosis, that's cell eating. Material here is taken up in form of solid. Cells specialized in this process are called phagocytes. A very good example of phagocytes are the white blood cells that are responsible for the body defense mechanism and are said to be phagocytic, e.g. white blood cells which take up bacteria by phagocytosis. The sac formed during phagocytic uptake is called phagocytic vacuole, as you would see in that diagram. That's your phagocytic vacuole. Then also pinocytosis is a form of endocytosis. In this case, cell drinking, the cell is taking up materials in liquid form. Vesicle forms here are extremely small. You can see in that diagram the vesicle is smaller than what you have for phagocytosis. But when you talk endocytosis, it is bulk transport, but in this case, it is an active process because the cell is going to expend energy. The cell is going to expend ATP. Then talking about exocytosis, it is the reverse of endocytosis, where endocytosis is intake. When endocytosis is intake, exocytosis is release. So when you talk about cells, let's say cells that releases enzymes, obviously because they are going to release the enzyme into the blood stream, so you are talking about exocytosis here. So exocytosis is the reverse of endocytosis. It is the discharge of materials, secretion of substance from vesicles of the cell surface. Look at what you have here. You can see those are secretory products. The vesicle just attaches to the cell membrane and finds a way to release its materials to the outside of the cell. Take for example, undigested remains from phagocytic vacuoles, undigested remains from phagocytic vacuoles, or useful materials such as enzymes are released to the outside of the cells in this way. You can see it in that diagram. Also, look at these other diagrams still on phagocytosis. Look at this other diagram. You can see how this happens. Materials, in a 6-step process, you can see small particles are taken up by phagocytosis to form phagocytic vesicles. A nearby lysosome, lysosome is the digestion center of the cell. So a nearby lysosome approaches the vesicle. Third, the lysosome fuses with the vesicle and discharges its enzymes into it. You can see that. 4. The lysosome enzymes digest the particles and product of digestion are absorbed into the surrounding cytoplasm. Now, 5. Digestion is complete and the vesicle moves towards the cell surface. When it gets to the cell surface, the vacuole membranes fuses with the plasma membrane and any undigested matter is voided, is released to the outside. So that's just a diagram to show you what happens during endocytosis and exocytosis. And with that, we have come to the end of this topic, talking about movement in and out of cell, or what we call cell and its environment, or more specifically transport across the cell surface membrane. And that just ends up everything about cytology, everything about the cell. So next up is going to be for us to take UTME past questions that relates to everything we have done on cytology, and then subsequently we will be talking variety biology, talking about taxonomy or classification studies and looking at the various groups of plants and animals. For myself and the entire team, it's bye for now.